Great. Okay, thanks very much. So, yeah, I also want to thank the organizers. I too am not, I don't work in molecular communications per se. I'm a synthetic biologist, but a lot of the work I'm going to be talking about is perhaps would provide some of the technologies and functionality we might need to interpret some of those molecular signals. So to give you some context, see if it's going. My group is really interested in this idea of reprogramming biology across scales. And we have kind of three major strands within my group. One is around what we call synthetic genetic parts and circuits. So these are kind of the elements that we build a lot of our systems from. And they provide us with the control over what happens within, say, an individual cell. So we can start to harness maybe innate biological capabilities of cells in new ways, or perhaps augment a cell's capabilities in a completely new and weird and wonderful way. Another strand of my group looks around how you can scale up some of those functionalities beyond, say, an individual cell to kind of multicellular collectives. And so that includes looking at kind of, populations of cells, how they communicate, and how we might control some of their, for example, spatial organization over time. And then the last kind of strand in my group at the moment is around trying to understand how evolution can be used potentially for engineering some of these systems, and how evolution itself is going to affect how these systems actually work. Now, I'm not going to talk about that at all today. I'm going to focus on these first two bits, and I'm going to try to give you an idea of how we actually go about programming cells with some of these circuits, some of the challenges and tools that we've built to kind of make that a little bit easier. And then I'm going to end by talking about a multicellular system that we're still in the process of actually building, but it might have some relevance or interest to some of the people here in the artificial life community. So to begin with, I'm going to focus, like I said, on these synthetic genetic parts and circuits. And the idea is that we can basically synthesize pieces of DNA now that we can put into cells. And by what's actually encoded on that DNA, we can affect what genes are expressed. And that will ultimately affect the phenotype or the behavior that that cell actually exhibits. Now, when we put that DNA into a cell, it ends up being executed by the cell. So the DNA itself is first read by these molecular machines called RNA polymerases. So these effectively transcribe the DNA into RNA, and that RNA is then converted into proteins by a ribosome. Now, this is a really complex and intricate process. And I think this diagram or this image kind of shows just the kind of how kind of crazily complex an individual cell is. And so the thought of trying to program this is like very, very terrifying to me. And so what we typically do is we abstract away a lot of those details, as you do, and we focus specifically on whether we can control how the DNA itself is actually being read. And so to do that, we take our DNA molecule and we use small sequences that we know have specific simple functions that we can piece together. So we introduce sequences called promoters, which effectively are a signal for RNA polymerases to bind the DNA and start reading it. And these RNA polymerases are shown by these little red blobs that are then producing RNAs, which are these kind of gray strands that are kind of emanating from the DNA. And then we can use other sequences, such as terminators, which effectively tell the RNA polymerases to stop transcribing the DNA. And so by placing these very basic sequences at different parts within a piece of DNA, we can control which parts of it are being read and the flows of these RNA polymerases across the DNA. Now, if we encode other sequences that have other functions within the DNA that's being read, then, and if those actual components affect, for example, the initiation rate of other promoters within that system, then what we can have is a situation where the flow of RNA polymerase in one part of the DNA is actually affecting the flow in another. And that can be used to build some very simple computational operations, such as an inverter or a NOT gate in this example. And the real key point here is that because we're using this common signal of RNA polymerase flux along the DNA, it means that we can connect together these components very easily by just making the output promoter of one of our gates the input promoter of another. And that's really what the field has done a lot over the last kind of 1 or 2 decades. And it's allowed us to create kind of simple regulatory circuits. So this is what's called a repressilator. This is effectively, it's a 3-node network where you have each of the nodes repressing the next in a cycle. And that leads to sustained oscillations in gene expression, which you could use as a kind of a clock you could imagine inside the cell. Now, we've got really good at building, like I said, these small, relatively simple circuits. But the real challenge that we face is that these don't offer sometimes the intricate computations that we might need to implement for certain applications. And that's really kind of hampering kind of progress in the field. Now, one of the reasons for that is that as we start piecing many of these components together, there are often lots of new types of failure mode that kind of crop up in these systems. And this kind of cartoon is showing some of the common ones that we see in my lab. So, for example, one of the failure modes we actually see fairly regularly is the fact that although we take these sequences, often we mine them from genomic databases that we have nowadays, and we think, for example, that this blue thing here is a gene, a coding sequence for a protein. Often those sequences have cryptic sequences within them, so they have multiple functions that we're not always aware of, and that can lead to, for example, flows of RNA polymerases in directions that we don't want, so they can actually interfere with the function that we're engineering. Sometimes parts just fail. This is really common, and I really feel for my students sometimes. There's a lot of biology we really don't understand, and so sometimes we can just use a part which we think should have a certain function. It just doesn't work in the context of the system we're building. Another type of failure mode, which is also very common, is what we call kind of sequence context failures. And that's where we might be using an identical sequence at different parts of a circuit, but because the sequences near that sequence are different, they can actually affect the function of that individual part. And so you end up with each of those parts having slightly different behavior. And that obviously makes it very difficult to build models or mathematical models of these systems for design, because you have to take into consideration all of these contextual effects, of which there are many. And then the final effect that we can't ever get away from really is the fact that we're putting this DNA into a living cell. And so we're reliant on a lot of the internal resources that the cell has, and that places a burden on the endogenous processes that are there. And so often there'll be uncharacterized interactions from the host cell to try and turn off the thing that we're actually introducing, because it has an impact on the fitness of that individual cell. And so it wants to remove that such that it can grow faster. Now, this is just a handful of a few of those problems, but hopefully you can see that as these circuits get bigger, the chance of seeing some of these things kind of increases. Now, these sorts of failures in engineering aren't uncommon in other fields. So if you're building, say, an electronic circuit, you might well mess up when you're building it. But the difference from, say, electronics and biology is the fact that in electronics, we often have tools that allow us to peer inside that circuit and understand what's happening inside in terms of the internal states and components, using things like, say, a multimeter to measure voltages and currents. But in biology, we don't have that luxury, and often we're only able to measure a very few states externally as an observer. And so one of the things that my lab's had to work on over the last kind of 5 or 6 years is building what we call a genetic debugger, which uses RNA sequencing, which is just a technology that allows us to read these gray strands that are being produced and calculate what we call transcriptional profiles, which is just basically a distribution of those reads across the DNA to try and infer indirectly these RNA polymerase flows to understand how these systems are actually functioning. Now, to give you a bit of kind of a better understanding of what that actually means in reality, this is an example of a simple logic or decision-making circuit where we have these 3 inputs, which are effectively small molecules that we can introduce, and we have sensors that we connect them to. And then we're using a fluorescent protein as our output, so we can look to see how yellow our culture is as to whether it's on or off. Now, using some of the parts I kind of mentioned and rules that I spoke about, we can effectively compile that logical description into a piece of DNA, so a DNA sequence. We can put that into a cell, and then we can see how well it works. And what I'm showing here is basically in each of the columns, the 8 different combinations of inputs that are available to that circuit. And in the gray distributions, I'm showing the levels of fluorescence, which is either low, so they're off, or high if they're on. And overlaid on that in this red and blue lines, there is basically the model that we use to design that circuit. So if everything's working, those two distributions should be overlaid. Now, in some cases, it's working pretty well. I'm pretty happy with those first 5 states, but as you get into these latter states, you start to see there's quite large deviations between what we think should be happening and what's actually happening. But the challenge is that we only can observe this yellow fluorescent protein, this gray distribution. And so we have no idea of knowing what's going on inside here and why it's not doing precisely what we think. And so what we can do is we can carry out RNA sequencing for each of those different states and generate these transcriptional profiles, which provide us with information about how much transcription, how many RNA polymerase per second are flowing across the DNA at these different components within the circuit itself. And this then gives us a much more holistic view of what's actually happening. Now, I'm not going to go into all the details here, but there's one interesting feature that kind of highlights some of the problems I spoke about before. So one of the things you'll notice is sometimes there are these like funny jumps that you see in the profiles. And these are actually cryptic promoters, which are embedded within these proteins, but we didn't know were there because we're not typically able to observe this kind of level of detail. So already we're starting to see things that we didn't realize were actually present. But in order to make sense of this very complicated picture, what we can do is we can zoom into one of these transcriptional profiles, and we can then interpret changes in the RNA polymerase flux based on some simple mathematical models to actually characterize how these basic elements that make up our computational devices, these promoters, are actually functioning, so the actual increase in RNA polymerase flux. And for terminators, we can look at the fractional change or the probability of termination in order to kind of characterize how well those terminating elements are actually working. Now, that obviously gives us kind of the lowest level information about how these individual parts are working. But to understand if the actual computational elements within those circuits are functioning as we expect, we need to collect together all of those measurements for the different combinations of inputs, and then we can start to collect them into sets which make sense for the various devices that make up our system. So for the sensors, that means collecting, for example, all of the states where one chemical is present or absent. And so here you can see there are 3 different sensors here, but we use them in 5 different places. The actual expression level of a promoter, which acts as an input for when that chemical is absent or present. And for the computational elements, we can actually start to draw out an actual experimental measurement of the response curve that those particular elements have and start to compare them to the actual expectation in terms of the models that we use to actually build these systems. Now, in theory, again, these things should be overlaid with each other. In reality, when you work with biology, that's never the case, but there are sometimes big deviations which often point to important points of failure. So here you can see my pointer? Yeah. So here you can see there's a big deviation between what we're expecting to happen with this sensor and what's actually happening. And this actually ends up being the root cause of failure in the system, which is actually at some point over here, which is obviously we wouldn't necessarily just randomly be able to figure that out. So this provides us with a lot of insight into what's actually going on. Now, as I mentioned at the start, this process of expressing these circuits isn't just a single step. There's multiple steps involved, multiple components involved. And so I'm not going to go into it, but one of the areas that we've been working on more recently is to kind of expand the capabilities we have in my lab to really unwrap the complexity that's there to give us measurements of all of these components and processes, which is really valuable when we're starting to build models of how these systems are actually working. So this provides us with the kind of the low-level information we really need if we want to build models that are predictive enough to actually reprogram cells effectively. So it's a key step, I think, in terms of getting to a point where we can reliably reprogram cells in interesting ways. Now, for the last bit of the talk, I wanted to talk about something completely different that we're also working on. So I come from a background in complex systems, and I've always been very interested in emergence. And so for the last few years, we've been also looking at ways of kind of controlling or working with living collectives. And one of the reasons for wanting to do that is that a lot of really interesting biology doesn't happen at the level of an individual cell. It happens at the level of a collective or a population. So things like bet hedging or swarming or learning or decision making, they're often an emergent property of the population itself. But studying these systems can be really, really challenging and often really costly in the lab. And so one of the things that we've done over the past few years is to build a kind of a very cheap 3D printed bits off the shelf, as some Raspberry Pis in there as well, and various optics you can buy from Amazon. A system we call the DOME, which stands for the Dynamic Optical Microenvironment, which embeds a number of different modules that are really useful for looking at collectives. So there's a sensing module, which is basically just a microscope, a kind of a fairly rudimentary microscope that can image cells that are present, or a population that's present in this sample here. But we also have an actuation module, which allows us to not just observe what's there, but actually interact with that population using light. So light's great because you can buy these little micro projectors and actually, with very fine precision, spatially temporally pattern that arena and interact with those cells in really interesting ways. We also embed some computation on this device. There's some Raspberry Pis that allow us to also potentially implement feedback control rules to maybe observe what a population is doing, make a decision on how to interact with it in order for some, I don't know, global population level feature to actually emerge. Now, as a proof of concept, just to see how well this system works, we've been playing with basically some pond life. So this is a variety of different organisms that you will literally find in a puddle or in a pond. And the reason we chose these is that they're motile, so they move around. So we're very interested in kind of swarming type behaviors, spatial behaviors. But they also do have innate responses to light, but those responses have not typically been very well characterized. So the DOME offers us a really unique opportunity to characterize how they respond to light in a very precise way. And that is specifically what we've been doing over the last year or so. We can take some of these cells, we can put them onto the DOME, and then we can basically zap them with different forms of light, different intensities, different patterns, both in space and time, and we can see how they respond. And so these videos, well, they're nice to look at, they're also really data rich. So we can start tracking each of these individual cells and get a lot of information about how those populations are responding, but also build effectively kind of digital twins or kind of a digital implementation of how the individual cells themselves also function and respond to light as an input. And one of the things we've been starting to do is to use that kind of digital representation of these microorganisms to build control laws. And so in order to do that, we need a very, very good model. And so this is just a demonstration of some of the aggregate data that we get from the DOME. So here you can see time series effectively, and these red patches are where we're just illuminating the entire arena. The blue lines are kind of the average of the population in terms of how it's responding. And you can see in terms of their speed of movement, which is on the y-axis, that they actually will respond to light by, in this case, slowing down. And that response is adaptive as well. So after a certain period of around, I think it's 10 seconds, they'll actually adapt to that light period and start moving again. So some interesting characteristics we can use here to actually parameterize quite a detailed model of how that organism is actually working. And to show that this does actually work, this is hopefully, I'm not sure if it should come out. You might have to squint. We're still working on this, but this is an example where we're trying to use that kind of single cell. You can see it. That's always a good sign. If people start laughing, it's like, oh yeah. So my lab's called the Biocompute Lab, and I hope you can see BCL on here. But the really cool thing here is that we're obviously able to use this system as a kind of a feedback control, almost like a cyber biological system we spoke about. So you've got the computation that's being done externally to the microorganisms, but the microorganisms are responding in a specific way to that input. The other really nice thing about this is that this isn't there's a lot of interest in synthetic biology around patterning, but a lot of that patterning is often very static. So that it might be that it's almost like photography. You'd have a film of bacteria, for example, and you'd shine some light on them, and the bacteria would differentiate in different ways. And then you take the light away, and then they have that state fixed for life. That's it. They're either a red or a blue bacteria, and that's it. In this case, this is effectively an active material. So you have this beautiful capability to dynamically change over time. And that's why you can see this sudden formation of this and then this dissipation of it as well. So this opens up some really interesting opportunities for dynamic control over time, which you wouldn't necessarily have in those other systems. And I think for some of the talks I've been to, I think these sorts of platforms are really valuable for actually trying out some of the interesting ideas you have with real living stuff as well. The only other thing I'll say is that, like, my group engineers a lot of microorganisms, and so we potentially also can get the cells to respond in very specific ways to light, and that's kind of the next step, is using this as kind of an optogenetics platform for with engineered microbes that can then carry out some of these tasks in a more efficient or quicker way. So that's everything I wanted to talk about. So in terms of take-homes, hopefully I've shown you that some of the work that we do in terms of reprogramming cells is really focused around controlling some of the flows of molecular machines that go on inside cells when genes are actually being expressed. I showed you that we use sequencing as a way of actually giving us a much richer understanding of what's happening inside these circuits. Biologists are absolutely obsessed with GFP for good reason. It's an amazing tool, but it's very limited in terms of I guess the dimensionality of the data you get from it. It's very good temporally, but it's very bad in terms of the number of internal states you might have within a system. So thinking about being a bit more creative about the methodologies we use to understand these cells, I think, is really important. And then finally, hopefully that last bit showed you that actually a lot of these experiments, sometimes when people talk about lab work, they get really scared about how expensive this stuff is. This system as a whole was around, I think, £700–800 to put together. And we can buy a lot of these microorganisms from places that sell these things to secondary schools for doing kind of experiments in the classroom. So you can, with relatively little money, you can actually start to have a platform where you can start to play with real biology in some really interesting ways. And hopefully that'll inspire some of you to maybe have a look at that. Like I said, my group works on a lot of different things. This is really only the tip of the iceberg, and I would love to have a chat to people. If there's anything on here kind of takes your fancy, let me know. So we look at things like, for example, the structure of the chromosome in cells and the way that that's used for regulation. Like I said, we're doing a lot of work around trying to engineer evolutionary paths. So not necessarily specifically getting a cell to mutate in one single way, but producing kind of distributions of mutations that are controlled in some manner. And we do a lot of work around genetic logic and other sorts of circuits inside cells as well. And with that, I don't do all the work all these people do. So I want to thank the students and the funders, and I'd be happy to take any questions.